This is Dolly. She is the first mammal to be cloned from an adult cell. She is also the world's most famous clone. Clones have the same DNA sequence as its parent. This makes them genetically identical. Clones are all around and are fundamentally no different than other organisms. There have been other clones made in a lab before Dolly that were cloned from embryo DNA. Yet Dolly was cloned from an adult cell. The birth of Dolly was a major scientific achievement because it shows that DNA from an adult cell, even though it had already specialized into one particular type of cell, can be used to create a completely new organism. One might wonder why scientists would clone a sheep. Well, Dolly was part of a research program at the Roslin Institute where scientists were looking into producing medicines in the milk of farm animals. After a great amount of research, scientists managed to transfer human genes into sheep and sows. The human genes that were used produced useful proteins so that, for example, the scientists could make the blood clotting agent factor, IX, to treat hemophilia. Being able to insert these genes into animals can be very difficult and can take a very long time, but with cloning, a scientist only needs to insert the genes once. This is because when an animal is cloned, the exact genes from the one animal with the inserted genes are copied and moved on to the other clone. According to animal research, Dolly was the only lamb out of 277 attempts to be born. Most animals that have been cloned before Dolly were cloned from an embryonic cell, but Dolly came from an adult cell, which is much more difficult to do. To produce Dolly, the scientists from Roslin Institute in Scotland used an udder cell from a 6-year-old Finn Dorset white sheep. Since the udder cell was an adult cell and was already programmed to make more udder cells, the scientists had to find a way to reprogram it so that the cell would make something else. To reprogram the udder cells, they had to keep the cells alive, but they also had to stop them from growing. The researchers were able to achieve this by altering the growth medium, or in simple terms, the soup in which the cells were kept alive in. Once the udder cell had been reprogrammed, the scientists injected it with an unfertilized egg cell. The egg cell had its nucleus removed, so the udder cell would be able to fuse to the egg cell. The scientists did this with electric pulses. When the nucleus from the adult white sheep cell, the udder cell, was finally fused with the egg cell, the research team had to make sure the resulting cell would develop into an embryo. The resulting cell was cultured for 6 to 7 days to make sure it divided and developed normally. Once the researchers confirmed that the embryo had normal development, it was transferred to the surrogate mother. The pregnancy went without any problems and was the only pregnancy that had went full term. After 148 days, the 6.6 kilogram white-faced Finn Dorset lamb was born. According to the Roslin Institute, unlike many other cloned animals, who usually had neonatal problems at birth, Dolly was a normal, robust lamb and was standing and suckling unaided within minutes. Dolly might have started her life in a test tube, but she lived a very pampered life at the Roslin Institute. The research team wanted Dolly to have a normal life, so it was decided that she should be allowed to breed and hopefully have her own offspring. Her first lamb was Bonnie, and she was born in the spring of 1998. The next year, Dolly had twins, and then the next year came triplets. Dolly producing normal offspring in the normal way showed that cloned animals could reproduce. However, a year after she had her triplets, Dolly's life would start changing. Dolly had arthritis in a hind leg joint. According to the National Museum of Scotland, this discovery added to the suspicion that cloned animals were destined to age prematurely. However, the cause of the arthritis was never determined. To make things worse, a virus started going around and infecting the sheep. This virus was sheep pulmonary adenomatosis, or SPA. This disease is caused by a virus that causes tumors to grow in the lungs of affected animals, and it is incurable. SPA is common in sheep raised indoors. Dolly remained healthy until February 10th of 2003, when an animal care worker reported that she had been coughing. Cloning of Dolly has opened new doors in the world of science. It has led to scientists producing more clones and has helped them make more reliable and easier ways to clone animals. Thank you for watching. I hope you enjoyed it.